Hello students, welcome to EPG Pathshala. I am Indira Ghosh from JNU and today we are going to talk on the module Metabolic Modeling in Biology under the paper called Computational Biology. Now metabolic modeling, why it is required? So our learning objectives is why we require to do it and what is metabolic control analysis, what is the flux balance analysis, and I will try to bring example in either cases and elaborate to you or discuss with you how to do these things. Why is started with this concept that it's no longer every one point of enzyme you design and find a inhibitor for it and you are expecting the results will be preferentially giving you a drug. Most often it has been failure. Earlier, we did not understand why the failure. Today, we are in a position to, thanks to genomics and proteomics and metabolomics also, we are able to understand that if we have to approach the disease and cure it, we require to have an integrated approach instead of reductionist approach. As this picture gives you a very clear idea that to analyze any of smallest tissue, you start backwards. So any system to recover disease, you have to start from DNA, then interaction and proteins, then interaction and the pathway, physiology of the system, and then you find out how to eliminate the disease. Hence, we have to be considering this as a very important starting point when we discuss this. First thing is biochemical kinetics and its systems perspective, and this has been taken, many of the concept has been included from Ethel Cornish Bowden's book called Fundamental Enzyme Kinetics. Anyone is interested to go in detail, please read these details in the chapter from 1 to 6. So before going to biochemical in silico biochemistry, we must revive our concept of what is biochemistry. See, what is the difference of makes it these enzymes? Why this is a very important thing? Because catalysis is a very simple chemical enhancers in chemistry. So what is enzymes do and why they're needed is shown in here, and in fact the reaction is always this reaction of carbon dioxide becoming in glucose degradation or glucose oxidation is a minus delta G. That means it's a free energy is negative, hence it should be happening all the time. However, it never happens all the time. The reason is between these two state, one is the glucose and that is reactant and the product which is carbon dioxide and H2O, there is a huge barrier which we call the transition state barrier, and this barrier height is the most difficult if one has to have from glucose degradation to happen spontaneously. This is one example, but all the reaction examples in biology are almost everywhere you have such kind of reactions. Very few would be spontaneous reactions. Major reason is, as we have discussed, these are all regulated behavior. In cell, nothing happens without regulation. The reason is it doesn't have spontaneity or any way. So all are controlled. Hence, at the level of biological chemistry, that is where the reactions has to happen, and otherwise the new product will not come up, or many of the macromolecule or molecules will not be made, which is necessary for a cell to survive, requires the importance or shows the importance of enzymes to lower this activation barrier. Of course, even if you increase the temperature, you can have it. However, in cellular system, actual cellular system, it is impossible to change the temperature. So obviously there are some thermophilic proteins which might be useful in this manner, but normal human being or the cell or a bacteria to grow, it is all room temperature or normal temperature. Hence, the enzymes are needed. Here I elaborate, enzymes are very specific because it will work for one type of reaction, not necessary for generic any other reactions. And also they always act at the room temperature, as we said. not between minus 0 or lower than 0 and higher than 100 degrees. There are few systems which might be working. However, it has been observed this kind of thermophilic or bacteria has a different kind of enzyme getup as well. So, just to revive your biochemical knowledge, we just want to say how is the kinetics and thermodynamics of the enzymes are done. If there is an enzyme which have a substrate S, then there should be ES complex. Actually, this is where the transition state barrier is skipped, and it becomes a product E plus P. Now, we know very well, we understand that it will be a single substrate and a single product. But you can have many reactants or many substrate as well. You can have many products. Now, what are these small K1, K2, etc.? They are actually association or dissociation and constants. However, basic premise of Michaelis-Menten's equation solutions tells us that the velocity of the reaction would be mostly governed by K2 and how fast the K2 ES becomes E plus P. Using this equation, we can go to the next slide. So, why to study in silico biochemistry? It is important we know that there are 2-3 types of biochemistry, but what it provides the detailed information on the genomics, proteomics, and metabolomics connectivity in the cell. That is how the functioning of cell really happens. Also, it gives us control picture of how to control the chemical flows and how it aids the cell growth. Changing the cell functioning is another topic which it helps a lot, and most important is that it gives us the essentiality of the particular enzyme which is required for cellular growth, etc. Now, to understand why we should study and what is the utility of this metabolic network and systems, the most important thing is that the important target should be having certain qualification. Number one is they disease-wise they differ. Like in cancer or infectious disease, one should abolish the activity of that particular major important enzyme or protein almost zero, not IC50. However, in case of metabolic disease, one should retrieve back around the normal, which is IC50 can do. And this is a different. So, we require to have modulator instead of eliminator in case of infectious disease. It effect may not be direct effect, even in some cases when there is a metabolic disease, whereas killing our organism is a direct effect. It might be a duration of therapy. We would like to have the therapy for a long, which for many disease like diabetes, we may take it lifelong therapy. So the drug to be effectively work for a long period of time with the human body. Whereas in the pathogenic bacteria, etc., or even cancer, killing the organism is short-term therapy can be adapted. One must understand if it is a genetic disposition, we can have different. So invasion is another. Immunity, failure of immunity or adaptation are the two other mechanisms by which the metabolic disease or infectious disease becomes resistant. Of course, there are environmental dependent and there are local tissue dependency. This makes the criteria being so stringent and different, it makes us to go for certain important issue which is shown right in the bottom. So, major one of these or the associated one, how to find them? Is that gene expression causes the effect of the disease? Can we remedy it just because the proteins can be lowered values? Can we do the transcription level or other level modulation? And can we do the inhibition at a controlled manner? Hence, all these questions can only be answered if we understand the flow of metabolite and understand the concept of ideally targets. Here I talk about target gene has to be very essential for the survival of the organism. It has to be located in a critical path so that they're connected with the survival. And most importantly, that vulnerability is the factor. That means this concentration of this gene inhibition must be in small alteration so that system becomes or organism become vulnerable. And that actually tells us to take care of the toxicity. So one has to understand that regulation, control, everything, and more than that, the vulnerability is important. Metabolic control analysis and whichever the topics which I am going to follow actually addresses this truth. Here I will take simple explain of if we delete or knock out a gene 100%, then inhibition of the enzyme is almost infinity. Now, if we level of expression, if we lower, that is 10 to 100 fold depending on the growth condition, the level of gene expression happens. And let us say we have three cases. One requires essential gene requires 5% activity to keep the cell viable. There are some which are very essential, but 30% of the genes is like this. And what could be the good target? The good target should be one whose minimum activity keeps the cell viable, will make it a terrible scenario. The reason is that you have to inhibit it to the 0%. Whereas best is the good target whose 50% activity should make the cell viable. That means if you inhibit it by 50%, you would achieve the goal. What happens in cellular system? Here I show you a plot where we have plotted with the same Km value, but two of them are competitive inhibitor and two of the plots are uncompetitive inhibitor equation using same equation but two different formulas. If we look at in vitro competitive inhibitor, which is the normally one finds, you see the 50% inhibition is achieved at almost at 0.5. However, lower than even 0.5 millimolar. However, if you look at the same in vivo, you would see the competitive inhibitor actually cannot achieve even 50% at the value of 0.2, which is almost 10 times more than the inhibition constant in situ, that is in vitro. However, the plots for uncompetitive inhibitor in vivo or in vitro are almost achieved within the same value of inhibitor concentration. So these plots are model plots using the functions bottom line shows you that probably competitive inhibitors may not be functioning so well in vivo, though they function very well in vitro. Most of the biochemistry done in vitro sometimes fails it because of the reason. And other way around, when it is uncompetitive, it gives you better idea. Another point I will discuss in the next slide. Well, different nature of essential targets. So let us say there are case 1, case 2 and case 3. Now case 99% target inhibition, that is protein inhibition, is required to show the cell killing. There are 90% and there are 50%. Now if we have the substrate concentration as it's shown, so what we require? To have 50% inhibition for the same target, 9 micromolar is sufficient. On the contrary, if 99% target inhibition is required to kill the cell, then you require almost 100 times more micromolar concentration. And this raises toxicity. Many of the drugs which ligand-wise perfectly suited and designed perfectly and biochemistry is done with a very good way fails because in cellular mechanism their inhibition is something like that. So the question is, can we find them as good target? So what we notice here that case 3 should be the good target. How to find these inhibitions? It will be wonderful if we can do these experiments in silico and then come up with the new idea. The plots of vulnerability plot, what I have shown here, which is available from previous data, what I have shown you earlier. We have to understand also that it is not just one enzyme or one pathway. There are several different kind of connectivity in the pathway. Okay, some are linear, some are forward branch, some are backward branch, some are polygonal. So how one can have a method, generic method to develop which will take care of all the connectivity. It is not only the connectivity. The next slide shows us how the diverse pathways exist in cell. It's not only connectivity, the function. On the left hand side, I have written the functions, that is what are the pathways are informed and biochemically identified. On the right, extreme right hand side, it talks about what type of metabolism it does. Carbohydrate metabolism, different lipid metabolism, protein metabolism, diverse system of metabolic system, and metabolites are produced by these pathways. Next slide shows you exactly a schematic picture from ECO-seq metabolic map. As you notice, all those kind of different connectivity is shown on the picture. Not only that, you will, if you carefully check, you will see some of the interactions don't have even a pathway. It is just standalone. How one can understand the whole picture, and that's the most difficult part. I will try to explain you how maybe in the first place that we would try to address only one part of the whole picture, and next part I will talk about the full pathways. So metabolic control analysis or a kinetic model build up based on a part of that previous picture what I have shown you. How we perform metabolic control analysis in a kinetic model. And this is the method in nutshell I'm explaining. And this is, first is, we require to do this control analysis. There, we must understand the quantitative measure of it. C is the control parameter which is shown, whereas J is the flux in that particular system. Then this is what we calculate, and this is delta E is the flow of the enzyme performance. Hence, we require to do some kind of methods to adapt for kinetic modeling. Detail would be discussed in the next. There are other two types of modeling. One is flux balance analysis, where actual values of controls, metabolic capabilities of the organism is measured, however, not as performed in every reaction mechanism. And another way is to look at, build up a single network and use the logical modeling where not any parameter but yes or no is used, like Boolean functions. I would be talking more about kinetic modeling and flux balance analysis in my following talks. To have the kinetic modeling, obviously we require the kinetic. So one has to understand the process of this kinetics of all the equations which is focus of our research. Hence, if we are looking at certain enzymes and their metabolite, how they are, let us say enzyme aldose works according to an order or uni-bi mechanism, then this will be the generic equation. The simple generic equation has been shown. However, we have to modify them accordingly. We have to compute the steady state. So how you calculate? There is a software available, and detail later on I'm talking, which uses mass conservation. And what actually means is this particular flux I, where I is several amount of all kind of reaction pathways. Then J is the particular flow of the substance. And hence, Cj is the upper super index and I is the bottom index. Total sum of all the flows should be equal to 1. That means, obviously, the total amount should be mass conservation to follow. The architecture. What we do, first we input the biochemical equation, which you specify your substrate product. Then you believe in rate equation, which you require many literature, whereas biochemical equations you can frame by bioseq. Now, then what you do is parameter of the rate equation, which is collected from Brenda and CCTBs, which is shown in bottom here. And then you simulate every reaction and retain your mass conservation rules. So there are two types of software possibilities, and you can, even some people have developed new software for doing this work. Slow detail of the flowchart, it says if we have a data from left hand side shows solve the Michaelis-Menten equation, then we require to get some parameters as I have shown you by schematic diagram earlier. What are the parameters are listed here. Similarly, once you have both parameters and the equations, you feed in and the references are given below, so which shows you how to solve this equation done. And this equation solving model equation, actually evolution of this model, this can be initial rate equation, starts with initial rate equation, and then you progressively solve the rate equations for at every time step. Here is an example how to set up your equation, and it is a metabolic pathway. Is a circle, a square inside white square is what we are interested. This is basically a glycolysis in parasite. However, one has to understand this is not independent, because entry of glucose is by something else. Phosphorylation is important criteria. So obviously ATP will come. So there are several inputs and several outputs also. So on the right hand side, the list of the proteins or the enzymes and the proteins necessary is shown here, and this paper is given here, is connects everything. So this equation says conservation constants are also used, because ATP cannot be just produced, so it should be constant values. So C1, C2, C3 are the constants basically used. Hence, the last is fourth conservation constant will be all the phospho groups in the glycosome that are not accounted by the entry of pi or exit of 3-PGA. So these few constant is only we can learn about it if we know the biochemical reaction. Hence, kinetic analysis becomes a little difficult. What we get out of it? We get out of it very interesting results, which has been elucidated here. The greatest share of flux control in this residue is found to be glucose transport state. Hence, the most interesting point here is, so if we have to stop this good target, it has to be only stop the glucose transport. If we really want to kill the trypanosoma, this is what we should address, not any other enzymatic pathways. And this is a very interesting development, because using other than glucose transport step, many other enzyme inhibition work was done in Glaxo for almost 10 years with meeting it with failures. The reason was that biochemistry of this was not really modeled earlier. Summarizing what we did earlier, here I have given you many of these tutorials to learn by yourself and many references. Another interesting example has been quoted, which is followed here, a very nicely articulated article from this. And one example only I am saying to show you how important to do this kind of calculation is artemisinic acid, which is the starting point of artemisinin, which is normally extracted, found to be produced in very common yeast, S. cerevisiae, by using metabolic control analysis, and produced enough so that it can be used as a simple drug candidates. As I completed your about the MCA or metabolic control analysis, there is another method, as I have mentioned in the beginning, that's called flux balance analysis. This type of analysis takes care of genome-wide understanding of the flux. That means here we are attempting to analyze whole of the system, total system. And how to go about, I will talk in few slides next. I would describe few steps, stepwise few slides, which will take care of the flux balance analysis. What is the flux balance analysis? Here also we follow the mass balance, but in principle, what we are observing in not solving every equation, but what we are trying to see is the flow of metabolite. In MCA, we are trying to understand the mechanism of interaction of the enzymes and how to inhibit them. What is the effect if we do the inhibition, competitive or uncompetitive? However, here it is not so much detail, but this gives you the great preferences of doing whole metabolic pathway analysis at one point of time. Because if we know all the metabolites, we just follow the flow of this metabolite. How we follow the flow. Now, if the metabolite is flowing properly or normally, then growth of a cell should be considered as 1. If it is not and terminated any in between places, then the growth of a cell should be 0. Keeping these two basic limits, one actually shows how to do this flow, study the flow. Well, this formulation is the prediction for formalism is this prediction of rate of reaction in a metabolite network, basically in natural state and, as I said, in a part-of state. So, part-of state can be knockout or knockdown or something of that kind. The best advantage is, as I told already, we do not need any prerequisite of kinetic data, which was a must for MCA calculation. So, if I have to compare or compare it with other type, metabolic network is a localized one-part network which is shown on the left-hand side, and flux balance analysis is a global network. So, two pictures are taken from the web link here, and you can very well understand that some information what you get from metabolic network control analysis is much more exclusive and more accurate on the topics of enzyme or inhibitors as you would like to focus on. However, flux balance analysis gives you a picture of set of enzymes or many pathways which are highly important for growth of the cell, which are not so important of growth of cell. So, two types of approaches gives us the facility to understand the topics in two different ways. Construction of this model is the step 1, which is the definition. Please note, I have given you the source from where I have adapted each of these slides. So you are most welcome to read all that as well. Let's begin with, this is the simple equation. Now let us have the three prop things. That is, as if B1 enters and something happens and A is your set, and then it goes to B, and there is also A becomes C. So, and after that B3 comes out. So if we have, even B1 comes out from B. So if we have a system like this, how we calculate? Now we calculate that these blue colors are the node, which are the metabolite species. Red colors are the age by which it is connected, and the black ones are input and output. So we can have some exchange which is inside, or we can have external flux. So we set up like this equation. Once we set up like that equation, we can formulate the actual differential equation of it. The change of A would be influenced by how much V1 enters, how much V2 rate, and how much is V3 comes out. And if we have set up those three equations, in case of here it is three of them, we can as well build up a matrix like S, and on the right hand side we will have the B. So basically we build up equation dx dt equal to S, and this S is called stoichiometric matrix. So stoichiometric matrix is something which ratios between each of these ingredient or each of these metabolites, and either they are present or they are not, either they are produced or they are inputted. Having this set equations and solving these equations, as we see, we go for the third stage, and this mass balance is steady state. And this dynamic mass balance at steady state, why we do the steady state? The reason is role of this internal all metabolites is to rate of their utilization. It is assumed in this kind of calculation, and simple assumption though, and is very valid, that is, nothing is wasted in a cell. That is, cell does not produce something which it doesn't utilize or doesn't excrete out. Hence, we have assumed that should be correctly understood. Hence, whatever comes in, it should have a steady state equation. And this assumption actually becomes very helpful for solving the equation. The reason is because it's steady state condition, we can equate it to be 0. And solving the equation becomes simple if you have this equation, and S dot V becomes 0 at the steady state assumption. But here comes the constraint, role of constraint. Okay. And that is the step 4. Because we have more number of reactions and less number of reactants or metabolites, or the reverse way, We always will have, as you know, in prokaryotes, you have many reactions. However, you have a less number of metabolite, because in prokaryote, it is utilized almost the same metabolite in 2-3 places, or some metabolite may not be used except one place or more than one. So in case of prokaryote, the situation is like this. In case of eukaryote, it might be other way around. But under every conditions, we have either the number of metabolite is less or number of equations are less. None of them are similar. Hence, to solve this equation, our number of set of equations becomes either larger than the parameters we want to. So we require some kind of intervention here to solve these asymmetric equations, or number of equations are less than number of unknowns, or the reverse, the number of knowns and unknowns are much more less than the number of equations. So one needs to use certain constraint. One constraint which is functionally perfectly okay is lower bound and upper bound. Obviously, every system works with a lower bound and upper bounds, and flux going into the system is considered to be negative. Flux leaving in the system considered to be positive. So this takes care of our most of these equations. So how we possibilities are done, like just an example, we have taken x plus y equal to 20, and maybe many values of x and y can satisfy the equation. Like we have x equal to 15 and y equal to 5, or x equal to 10 and y equal to 30. We can have many type of values, minus 10 and 30 and all these. So if we use y equal to 4, let us say, then only we get x equal to 16. So this type of principle applied to FBA for using the physiologically relevant concentration, such as bounds of exchange reactions, etc., so that we can narrow down the solution space. Optimal flux. How we calculate the assumption is metabolic network is optimized with respect to certain objectives. That is, as I told, system works optimally. That means we can use this what you call function z as something to do with how much it is produced, and minimize or maximize our equation. So z equal to equation like s dot v type equations, and do that. We can do any kind of reaction, this optimization, and techniques mostly used is linear programming problem. Sometimes people are developing genetic algorithm as well for this, but I'll just trick up myself that there are available modules which helps you to modify this meaning of flux distribution. How to choose the objective function? It is reasonable to choose the cellular growth. That is, the cell grows only when optimally everything is happening, and cell dies if optimally that thing doesn't happen. So even if one perturbation or one metabolite change might cause the cell to grow or not grow, we will come up with actual example. Here I sum up or show you what is the effect if we have some enzyme is knocked out. That means we get lower bound 0 and negative for reversible, and upper bound is positive value, something like that. Here I show you a very famous picture taken from the paper, where I just summarize you the steps, 1, 2, 3, 4 steps, and at the end you can see how the linear programming manages to balance between the z function. This is the starting of working computationally implement such a system for genome-wide understanding. However, it is very important to once more summarize yourself about the limitation or the points to be remembered when one does the flux balance analysis, which is shown in next slide. From the flow chart, you have understood the flow of the whole equation step by step. Now I'll talk about summarize the concept of FBA in a nutshell, as I said earlier too, but this slide will be very useful in the long run to keep yourself aware. Number one, this is a quasi-steady state. That means the matrix S is defined as stoichiometric matrix and V is the rate of the reactions. Next, there would be capacity of reversibilities written here in this slide. So AI should be varied. VI, that is the rate of every I, means every metabolite, should be varied between a lower and higher bound. Always it will be reversible. Of course, some reversible or irreversible reactions also can be managed to fix up. But reversibility is assured if its Ri is greater than equal to zeros. And we should have measurement or known rates as well to check it. And optimality is done by a linear programming, as I said. And what is the effect of one gene or one enzyme inhibited 100% is given by if J becomes the knockout gene, then we should set AJ equal to 0 and BJ equal to 0. And for VJ, we solve the equations. This is an example done and publication by Bud Paulson's lab in UCSD. And they took whole E. coli genome, they constructed all the pathways, annotated every sequence possible, set up the stoichiometric coefficients for all of them, and then optimize everything in silico. And what enzymes? They try to delete every gene deletion possible, and they try to see whether the system allows the growth to happen or becomes zero. In silico, gene deletion are shown here. As you see, these dark blue colors are shown very clearly that those mutations actually doesn't make any, Z mutant by Z, that means it is almost 100%. Whereas there are some of them which you can see on the left hand side, all of them being muted or stopping their flow, the growth becomes zero almost. So what it means? It only tells us that not necessarily, so almost 30 to 40% proteins actually doesn't affect the cell, normal cell. And yellow bars represent the gene deletion that reduce this biomass at around less than 95% in the wild type. So this is interesting. So one can understand which are not so important genes, and one can understand very straightforward which are the important. However, very interestingly, when they started looking at this was a study by one gene deletion. When they started having two gene deletion, they came out with a wonderful ideas. And I'll show you next slide that. What they figure out, the rerouting of the metabolic flux happens. Here I show you the black one is wild type, and fatter they are, that means they are flowing much more, and thinner they are least flow. Red is the mutant and blue is the mutant. Now two mutant, one is on the top, here I show on the top, one mutant is shown here on the bottom, and you can see that these things, three types of values are given here. So one knockout was ZWF. It just resulted 1% reduction of the mass biomass. However, when they did ZWF and PNT together, two of them, one is this ZWF is glucose 6-phosphate dehydrogenase, another is transhydrogenase. And these two are two different pathways, two different distinct topology pathway. And very interestingly, double knockout resulted 8% reduction. Hence, one has an opportunity of magnanimous calculations of different kind of combination. This establishes how robust this metabolic pathway is. Hence, it's not so easy to tamper one or two genes and get around the whole system to make it submissive to stress. This is a technique, though, has to be verified after doing experiment, which has been done many cases too. Summary of this particular study, as I summarize with FBA, it gives you metabolic genotypes for all the biochemical. You can do it different strain specific, you can do it different kind of it, and genome wide, all the metabolic pathway. That is the good point. And relatively fast, and it has a critical gene product. One result we already saw, only few genes are really responsible for central metabolism. Basically, they are the products. Also, it can verify our knockouts and design the growth experiment with respect to these knockouts. I come to the final idea, And this particular example give you the full picture that we are deviated from the reductionism approach of biology. That is, you just take one protein and get one gene out of it, or one gene and one protein out of it. We have now learned many of them can be possibility. Also, one protein to inhibit will achieve the systems to succumb to the pressure is not necessarily going to be true. Hence, the approach has to be away from traditional approach, that is reductionist approach, and slowly we are going towards the integrative approach and holistic approach, which people sometimes talk about. But mainly I would have been obviously trying to convince you that many of this work, many of these interactions actually makes the system to succumb. So drug finding, target finding, etc. has to be approached in a very multifaceted way. Slide shows the contains all the references given. So in summary, I would like to inform you that metabolic pathway is important to understand the phenomenological behavior of the organism, and many way to analyze these informations and understanding are there, which are described earlier, but in detail metabolic control analysis and the flux balance analysis is discussed elaborately. How to build up metabolic MCA and how to build up the flux balance, both have been discussed, how to build these models and their predictability, etc. Also, it has been discussed what techniques has to be used, what parameters has to be used, and where to find them. Comparison between these two also with the limited informations have been discussed. But most importantly, I have shown you two real case study. That is where the E.coli has been used as a model system, and another case was has been used is this trypanosoma glycolytic pathway. Hence, what we found also is very significantly important, which has been described. However, both the cases, target identification is the step where these methods would be utilized and applied very frequently in future. Thank you.